And let's jump into our Google Classroom here. There we go. Let's see here. Protein synthesis. Again, protein synthesis is just a fancy science way of saying making proteins in the cells, which is exactly what we've been talking about this week. So just as kind of a real quick overview of the stuff that we went over on Tuesday, we have genes in our DNA, and genes are segments of the DNA that has the code in it for a protein. So our DNA is huge. It's millions of bases long, and a gene is maybe a couple hundred or 1,000 bases long. And our DNA has lots and lots of genes in it. I think there's a couple hundred genes that scientists have identified in human DNA so far. And each individual gene makes a specific protein. It makes them out of these structures called amino acids that we string together and fold up into a specific shape, and that is what gives us our protein. So the main thing that we talked about on Tuesday is that DNA is in the nucleus where it's nice and safe and protected from being damaged, but proteins are made outside of the nucleus. They're made in these little protein factories called ribosomes. Hold on, where's the little pointer at? There we go. Proteins are made in ribosomes, which are not inside of the DNA. So we've got to copy the code from our DNA and carry it to the ribosome in order to get turned into a protein, in order for protein synthesis to happen, building proteins. So DNA is our original master plan copy. Our version that's copied and taken out is RNA. We talked about those three differences between RNA and DNA. You have your sugar that's different. The D in DNA comes from the deoxyribose sugar, whereas the R in RNA comes from the ribose sugar. DNA has 4 bases, ACG, T. RNA also has 4 bases, ACG, U. So again, there are no T's, no thymines in RNA. Instead, we have uracil, aka U. And then the other main difference, DNA is double-stranded. It's that double helix twisted shape, and RNA is single-stranded. So here is our double-stranded DNA that has thymine, and here is our single-stranded RNA that has uracil. There are 3 different types of RNA. rRNA, which we're not even really going to talk about at all. tRNA, which we're actually going to talk about in a little bit more detail today. But the big one, the super important one, is mRNA. This is the one that we focused on Tuesday, and this is what is carrying that DNA message. Because of that, it's called messenger RNA. That's where the M in mRNA comes from. So RNA copies the DNA code and takes it out to the ribosome. Again, remember that the big difference is that mRNA, the part that matches up with the A on the DNA is not a T, it's a U. So C and G still match up with each other. G and C still match. A on mRNA will match up with the T on the DNA side. But when the DNA has an A, the mRNA matches that with a U, not a T. So a protein called RNA polymerase helps out. It unwinds and opens up the DNA and then reads the gene and makes a copy of the DNA in mRNA. So again, the DNA T matches with an A. The DNA C matches with a G, the DNA G matches with a C, and the DNA A matches not with a T like it would on DNA, but it matches with a U instead. So we go through and we build this nice big long mRNA sequence that is a copy, it's like a photocopy of our gene. Only this one can leave the nucleus, go out into the cell, and match up with that ribosome, that protein factory. So that's kind of where we left it on Tuesday. We had the mRNA being copied, the bases matching up and then pulling off, and then the mRNA goes and leaves and heads out to the ribosome. So when it gets to the ribosome is in charge of figuring out which amino acids are supposed to go into the protein. Remember, the ribosome is the protein factory, and proteins are built out of amino acids. You take lots of amino acids, string them all together in a line, and then fold them all up, and they get turned into proteins. The way that the ribosome does this, the way it figures out which amino acid comes next in the protein, is through these sets of 3 bases called codons. So each set of 3 bases in the DNA, or then copied in the mRNA, is called a codon, and it contains the code, hence the name, for a specific amino acid. It's sometimes called the triplet code, since there's 3 of them, like triplets. And each set of 3, each codon, corresponds with one specific amino acid. So in our RNA, in our example RNA down here, we have 1, 2, 3, 4, 5 codons in it. There's 15 bases total. Separated up into groups of 3 gives us 5 codons. So in this RNA sequence, the ribosome would read it and say, all right, first up, we've got AUG. That's going to give us methionine. That's what this little yellow ball represents. Okay, next one, GUC. That's going to give us valine. Next one, AGU. That's going to give us serine. Next one, CCA. That's going to give us proline. And then finally, UAA doesn't actually code for a amino acid. It's what's called a stop codon. It just tells the ribosome, stop reading. This is the end of the gene. Don't need to go past this point. So that's what tells the ribosome to end it and turn that string of amino acid into a protein. Yeah, Ashley, go for it.
I was just gonna ask if the codes were always the same or if it's just like an example.
So the codes are, let me back up a little bit. Obviously, each mRNA is going to have a different sequence of codes. Most of them are going to be a lot longer than this. They might be dozens or hundreds of codons long. But each set of 3 bases codes for one specific amino acid. So like AUG will always result in a methionine being placed. CCA will always result in a proline being placed. GUC will always give you a valine. So in that sense, the code is always the same, meaning that a set of 3 bases is always going to code for the same amino acid. And in a little bit, we'll look at a big chart that shows us what every single possible combination of codons could give us. And that's actually what will be involved with your assignment for today. So we'll go over that a lot when we get there. So yes, every mRNA is going to be different and have a different set of codes, but each set of 3, each specific codon codes for one specific amino acid. So that's how our DNA code works. That's how we can take this long string of bases and turn it into a protein, into a structure that's going to go off and do something in the cell. So when I say that the ribosome reads the code and knows which amino acid to put, that is kind of simplifying it a little bit. What actually happens is that that codon on the mRNA has to match up with a structure called tRNA. So tRNA is this kind of funky looking RNA. It has what's called a clover leaf shape. And so really quickly, I'll show you what one looks like. So here is what a tRNA looks like. It's one long strand of RNA that kind of gets wrapped around itself and has these kind of bubbles or these leaves at different points. And a tRNA is responsible for carrying amino acids around the cell. So the amino acids aren't just floating around in the cell willy-nilly waiting to get used. Every amino acid is originally attached to a tRNA. So when the tRNA and the mRNA match up, that's how the ribosome knows that we've got the correct amino acid coming in. And the way that they match up is with something called an anticodon. So our codon is that set of 3 bases that's in our mRNA. The anticodon is a set of 3 bases on the tRNA that matches up with the mRNA. So if the mRNA has a G, the tRNA is going to match it with a C. If the mRNA has a U, it'll be matched with an A. And then C will be matched with a G. So down here is our anticodon. And up here is where the amino acid gets attached. So like how in this example over here, we represented amino acids as like these little balls. Imagine that there's a little ball that's attached to the tRNA up here. And as the amino acid, or excuse me, as the mRNA goes through the ribosome pulls in all these different tRNAs and they match up with the mRNA at the codon and anticodon. So for example, our first codon over here was AUG. The matching anticodon for that would be UAC, right? Because the U matches with the A matches with the U, and the C matches with the G. So if we have AUG as our codon, the matching tRNA is going to have UAC as the anticodon. That way, they can bring them together, say, yep, these match, and add the little amino acid up here to that growing protein chain. And in the video that I'll show you guys at the end of our class today, it does a really good job of showing exactly how this process works, kind of in motion. So again, if we have a mRNA down here that's moving along, we have a codon that comes up, and the codon reads ACU. So the ribosome goes, okay, I need a tRNA that matches with ACU. So it brings in this guy, and the tRNA has UGA. So A and U go together, C and G go together, U and A go together, So these guys match. So this is going to be the next amino acid that gets added on to the protein chain. So when the codon and the anticodon match up, the amino acid comes off of the tRNA and gets linked on to that growing protein chain. So again, we have this mRNA that copied our DNA code. It comes out of the nucleus. It goes into a ribosome. As it's going through, the ribosome is essentially reading the code and then matching the correct tRNA to the correct codon. It lines up the codon and anticodon. And if they link up perfectly, then it takes this amino acid and adds it to the amino acid chain that it is making. All right, does anybody have any questions so far about mRNA or codons or tRNA? All right, cool. So again, each codon is a code for a specific amino acid. So each set of 3 bases, each 3-letter code corresponds with a specific one of those 20 amino acids. So if you do a little bit of math, you can figure out that if there's 3 spots and there's 4 possible codons, or 4 possible bases for each spot, there's 64 different codons, but only 20 amino acids. So some amino acids might have more than one codon. So like a lot of times the amino acid will, as long as the first 2 letters are there, those will be kind of specific, and then the last letter can kind of be whatever. So like, I'll show you some examples in a second, but like CCC and CCA and CCG might all code for the same amino acid. So there are different combinations of codons that you can use in order to get the same result, the same amino acid. And then also there are some of those stop codons. So a lot of the ones that begin with U are stop codons, like UUA or what else? UAA, I think. Those tell the ribosome to stop translating, stop with the creating the protein. This is the end. This is it. We are done. So we could have lots of different codons for a specific amino acid, but no matter what you have, each codon will code for the same amino acid every time. AUG will always result in a methionine being added to the amino acid chain every time. So here is one version of a way to read the genetic code. So scientists have gone through and figured out exactly which codons line up with which amino acids. And so there's a whole bunch of different types of charts that are out there that show you how to read the amino acid code. So on this one, I really like, for example, it's a lot prettier than some of the other ones. Each codon is obviously made up of 3 bases. And so for this version of the genetic code, Your first base is your middle kind of ring. And then you follow that to a little bit more outer ring for the second base, and then the smallest ring for the third base. So, for example, with that AUG example we were looking at earlier, AUG is methionine. So the way that you'd read this particular code is that A is in this section right here, and AUG, AU is, so U is up next, and U is this area down here. And then G, AUG, G is right here. So AUG gives us methionine. So this is kind of a little bit more artistic way of looking at it, a little bit more kind of aesthetically pleasing version. A lot of genetic code tables look something a little bit more like this. And this one I specifically pulled right out of the biology STAR test. So in this one, it's less pretty, but just as easy to use. Your first base position is over here on the left-hand column. And then your second position, you go up at the top. And then the third one is over here on the side. So, for example, if we're looking at that AUG example again, we have our first one is A. So we know somewhere this row right here has all of the A starting bases. Our second one is U for AUG. So U is this one. So we know that it's going to be in the first column. So it's somewhere in here. And then AUU, UC, UA, AUG is MET, which is the abbreviation for methionine. So that is how you use a codon chart that looks like this. You find your first letter to get the row. You find the second letter to get the column. And then that particular spot is broken up by individual third letters. So like if we're going GGG, for example, G is down here in our first position, down at the bottom. Our second position, G, is over here. So we're all the way at the bottom, all the way on the right. And this is one of those ones that's easy because no matter what your third letter is, if your first two bases are GG, you're going to be glycine is your amino acid that you're going to put in there. So all 20 amino acids are represented on this chart, and lots of amino acids have different combinations of codons that can get you to them. So like leucine over here, anything with CU or any UUG or UUAs will give you leucine. We also have our stop codons over in this segment. So UAA, UAG, and UGA are all stop codons. Excuse me. But no matter what codon that you have, you're always going to read it in the same way. Find the row using the first letter over here. Find the column using the second letter up here. And then find where in that area your specific codon is located. So I showed you guys how to do AUG and GGG. But now I want you guys to help me out a little bit. So I shrunk this down a little to make it so I could put some of these questions in here. Oh, look at that. I already did GGG for you right up there. We said that it is glycine. So I want one of you guys to either out loud or in the chat, let me know if we have the codon UCA, which amino acid are we going to get? What's the amino acid abbreviation that comes from UCA?
It says here, there.
Mm-hmm. Yeah, SER, S-E-R, short for serine.
So we
Yeah.
have just to look at the letters and just look at it short, and that's it. Because
Yeah, using the short version is totally fine. But yeah, we have U, so we're up here. We have C, so we're right here. And all of our UC somethings give us serine. So UCA gives us SER, S-E-R, but so does CC or CG or CU. They all give us SER or serine. Nice. What about CAU? What amino acid are we going to get from CAU?
HIS.
Yep, exactly. C is right here. A is right here. And U is the very top box. So CAU gives us HIS, which is short for histine. Good, very good. All right, what about GCA? What amino acid is GCA going to give us?
ALA.
ALA, let's see. G is right here. C is right here. And yep, oops, sorry. All GC guys give us ALA. So GCA is right here, and it's going to result in ALA. And then finally, what about AAA? What is AAA going to give us for our amino acid?
LYS.
Exactly. LYS, which is short for lysine. So A is right here, and then A is right here. So they meet up in this one, and then let's see, AAA right here gives us lysine. Good, very, very good. So that is how you use this codon chart in order to figure out which amino acid you are going to get. Codons are always sets of 3, and so you use your first letter to figure out the row, the second letter to figure out the column, and then you can look where it's pointing you and find the amino acid that you are looking for.
Mr. Michael, I have a question, please. On the
Yes.
STAR test, they give us the same example like this one?
Yeah, I literally pulled this chart directly off of an old STAR test.
Okay, thank you.
So if you ever do take the biology STAR test, and if they have a question on there about codons and amino acids, it'll be a chart that looks either exactly like this or almost the exact same, with like the first letter over here, second letter up here, third letter over here.
Thank you.
Yep, no problem. Good question. Any other questions about the codon chart? Anybody else still a little bit confused about how exactly we're figuring out? Actually, wait, hold on. How we're figuring out which amino acids go here?
No.
Some more of this for your homework for today. So if you do have questions, now is the time to ask.
On the homework, it comes with the chart too for us to look at? Oh, okay.
Yep, exactly. All right. So that is how a gene in DNA gets turned into a protein. Because once we go through all of the codons in our mRNA, we're going to have a big, long string of, it could be a couple dozen, it could be a couple hundred of these amino acids, and then we wrap them all up in a certain way, and that is what becomes our protein. So here's sort of a super duper simple example or kind of overview of this. So this final process that we just talked about, where the ribosome reads the mRNA and the tRNA comes in and drops off the amino acid, that is called translation. So transcription was like what we talked about on Tuesday, where we take the gene of a DNA, copy it into an mRNA. Translation is when we take that mRNA, put it through a ribosome, and it gets turned into a protein. So this figure is just kind of okay. It has a very, very basic overview of everything. I like this one a little bit more. So we have DNA that has these different genes in it, and we want to copy this first gene and turn it into a protein. So we have our kind of template DNA strand. So that's like our starting DNA strand. We go through transcription and turn it, make a copy of it in mRNA. So again, on the DNA, we have an A. So we're going to match that with a U, not a T, because remember, always remember, RNA, no T's. T's are not allowed in mRNA or any kind of RNA. It has to be U. So U will match up with the A on DNA. The C's in DNA match up with G's. The G's on DNA match up with C's. And the T's on DNA will match up with A's. So we get this nice long string of mRNA that then pieces out from the nucleus and goes over to our ribosome, which is where translation happens. So our ribosome is what reads these mRNA strands in codons. So it goes 3 by 3 by 3 by 3. First codon, it says UGG, and it knows, okay, that means we need a tryptophan. Next codon is UUU. Oh, we need a phenylalanine. Next codon is GGC. Oh, we're going to need a glycine. Next codon is UCA. Oh, that means we're going to need a serine. So we take these individual amino acids, link them all together, and then fold them up, and they get turned into our protein, the final thing that we're trying to make. So all the stuff in your body that is controlled by genes, it has to do with genes making proteins that then do some sort of job inside the cell. So you may have heard of mRNA in the news over the past couple months because the Moderna and Pfizer vaccines that have come out for COVID-19 are the very first widespread use of mRNA vaccines. What that means is that scientists figured out the protein structure of the spike proteins, which are those little knobby proteins that are the little like things coming off of the coronavirus. If you ever see it in pictures, it's like the little ball with all like the little fuzzy things coming off it. Those fuzzy things are proteins called spike proteins. And so scientists figured out what is the amino acid sequence of a spike protein. They then, once they knew that, they were able to make an mRNA strand that would give you that spike protein. And this mRNA is what they inject into your body, inject into your cells. So the cells pick up this mRNA, turn it into spike proteins, and then your immune system attacks those spike proteins. And that's what teaches your immune system how to recognize and attack COVID-19 without actually having to ingest you with COVID-19 or without you being infected by the actual virus. You're just infected by mRNA that tells your cells how to make spike proteins. So that's a really cool modern application of knowing that this protein and this mRNA all work together in order to create these structures. So here again, this one is one of my favorites because it brings in our little tRNAs right here. So we have our DNA. We have this part of the DNA that is a gene that we want to make a copy of. We make our mRNA strand through transcription. That mRNA leaves the nucleus. A ribosome comes along, attaches onto it, goes down the line, starts to make this amino acid chain, and it makes it specifically by taking these weird tRNA guys, matching them up with the codons, and then the amino acid that the tRNA was carrying gets added into the chain. So as the ribosome goes along, it's constantly matching and adding and matching and adding these tRNAs and amino acids. And that is what is translation. That's what in science we refer to as the process of translation. The ribosome reading the mRNA and turning it into a protein. So I actually have that really cool video. We started to watch kind of the first part of it yesterday, and I have it posted up in the Google Classroom if you ever want to take a look at it. So we've got our cell, and we're going to zoom way into the nucleus. Oh, no, stop, go away. Remember, the nucleus contains the DNA, it's inside. Because if the DNA is outside the nucleus, it could be damaged by something, it could be hurt by something, and any damage to your DNA is very, very bad. We don't want that to happen. So we protect it by putting it in the nucleus. So all of our genes, all of our what's called our genome is hanging out in the nucleus. It's packaged into things called chromosomes, which we'll talk about a little bit later on in the term. And it's separated out into genes. So the gene is where we have the information to make a protein. So when the cell needs to make that protein, it brings in its buddy, RNA polymerase. And this guy is what matches up the RNA bases with the DNA template. So we're matching these guys up. G's go with C's, T's go with A's, C's go with G's, and A's go with U's. Remember, normally in DNA, there'd be a T here, but this is not DNA. This is RNA. So we have a U instead. Make an mRNA, make an mRNA. And again, once the mRNA, the bases get matched and it gets attached to a few more bases down the line, the mRNA immediately starts to come off. It doesn't need to hang around on the DNA all day. It's got stuff to do. So we go through, we read the whole gene, and we make a copy with our mRNA. So before the mRNA leaves the nucleus, stuff happens to it. Don't worry about that. We're not really going to talk about it in this class. But once it's all ready, it'll head out of the nucleus. So this is what is allowed to leave the nucleus and go be read by the ribosome, right? DNA is too important. We got to keep it safe. It has to stay inside the nucleus. But our RNA, this is just a copy. We can always make more copies. So the RNA comes out and heads over to the ribosome. So here we have our big ribosome structure, and the mRNA is going to go right through the middle. So to help us along, we have our tRNA. So here's our funky tRNA structure. It's got the amino acid attached up here on one end, and that anticodon that's going to match with the mRNA down here on the other end. So as the mRNA goes through the nucleus, you'll see a tRNA kind of like fly in, match up with the mRNA, codon to anticodon, and then the amino acid will be dropped off and attached to the chain that the ribosome is making. So we've got these tRNAs that are flying in, they drop off their amino acid to the chain, and then they peace out and leave. So they come in and match, they get moved down, and they attach their amino acid so that another one can come in, and then they get moved down again and depart. They go off into the cell looking for another amino acid to attach with. So we're growing and growing and growing this big, long amino acid chain. And once we get to the end, once we hit that stop codon, the amino acids come together, get folded up, and form the specific protein that we are making. All right, let me show you guys that video one more time without me talking this time, so you can see how everything happens without me constantly pausing to talk about it. Obviously, before we go there, I will say that this process actually happens much, much faster than we're seeing here. Down at the cellular level, things happen extremely quickly, extremely rapidly. And so they've actually slowed it down a lot just so that you can actually see what is going on here. So I would say that I'm showing this to you in real time, but in reality, this happens like they can read like dozens of tRNAs can cycle in and out a second as the amino acid is being built, as the mRNA goes through the ribosome. It's a very, very fast process. So we're going to take this gene and we're going to turn it into a protein. But first, we got to copy the gene to mRNA. So this is transcription, what is happening right now. So now we have our mRNA and stuff happens to it that we don't need to worry about. And it finally gets to leave the nucleus and go be turned into a protein by the ribosome. So to make a protein, we need amino acids, which are carried around on tRNAs. So the tRNAs are going to come into the ribosome, match with the mRNA, drop off the amino acid, and then be kicked out. So they match, They drop off and then they're kicked out. And then finally, once the ribosome reaches the very end of the amino acid of the mRNA and hits that stop codon, the amino acids are then folded up in a specific way to be turned into a protein. So we take our gene that was in our DNA, just a long sequence of ACTG bases in DNA, and we have turned it into a protein, a structure that's going to go out and do important jobs in the cell. And that's what this protein synthesis process is all about. Taking the information in DNA and actually using it to create something inside the cell. All right, so does anybody have any more questions about this transcription, translation, protein synthesis process? Anything else about this that
No.
you're still a little not sure about or that you want me to go over? Alrighty. Well, I hope you guys enjoyed learning about this process. To me, it's one of the coolest things that we talk about in biology. This kind of whole process is almost this whole puzzle that has to fit together piece by piece by piece in order to turn that DNA gene, that string of DNA, into a protein in the cell. I think it's really neat, and I hope you guys enjoyed learning about it as well. So for the worksheet for today, it's very similar to what I had you guys help me with some of our examples of the amino acids a little bit earlier. So I give you guys the exact same chart that we were just using on the notes and that you'll see again if they have a question about it on the STAR test. And I just want you guys to tell me, what do these 7 different codons code for? So you just have to put in the abbreviation that's here in the chart. You don't have to look up and write out the name of the amino acid unless you really want to. So AUG, CGA, GCG, GAA, CUG, AAG, and UCC. Use what I've taught you. Go through, look at the first, second, and final letters, and figure out which one of these 20 amino acids corresponds to those particular codons. All right, and then let me see if Monday. So Monday's question when we come back next week just has to do with that tRNA. So keep your eye out for this question popping up for your daily assignment on Monday. All right, does anybody have any final questions about genes, DNA, tRNA, ribosomes, amino acids, anything that I've talked about the homework or about these notes so far tonight?
No.
All right, thanks, Naya.
You had already answered the question I had asked you before the class started.
Perfect.
Thank you.
Good to know. No problem. No problem, Samyan.